All right, guys, welcome to the Chapter 17 lecture about blood gases, pH, and buffer systems. Now, we've broken down Chapter 17 into two parts because it's a pretty complicated chapter. So what we're going to cover in the first part is kind of what the blood gases consist of, how pH is buffered, what that physiology looks like, and then we will move into part 2. I'll be posting that at the beginning of this week as well, probably tomorrow or the next day. And that will cover more whether someone is going through respiratory acidosis or metabolic acidosis or alkalosis. So it'll be more what you're going to see as far as the lab work goes, and then being able to surmise a diagnosis from that. Okay, so let's just go over the objectives that we're going to cover today and in the next lecture. So after completing this chapter, you should be able to describe the principles involved in the measurement of pH, PCO2, PO2, and the various hemoglobin species. You should be able to outline the interrelationship of the buffering mechanisms of bicarbonate, carbonic acid, and hemoglobin. Explain the clinical significance of the following pH and blood gas parameters: pH, PCO2, PO2, actual bicarbonate, carbonic acid, base excess, oxygen saturation, fractional oxyhemoglobin, hemoglobin oxygen binding capacity, oxygen content, and total CO2. You should be able to look at data and determine whether it's normal or represents metabolic or respiratory acidosis or metabolic or respiratory alkalosis using the Henderson-Hasselbalch equation and blood gas data. Then from there, you should be able to identify whether the data represents uncompensated or compensated conditions. This is what we're going to cover in the next lecture. We won't be covering this quite yet today. We're going to identify some common causes of non-respiratory acidosis and alkalosis, respiratory acidosis and alkalosis, and mixed abnormalities. Then you should be able to state how the body is going to attempt to compensate for that using the kidney and lungs for various conditions. Describe the significance of the hemoglobin oxygen dissociation curve and the impact of pH, 2,3-DPG, temperature, and PCO2 on its shape and release of oxygen into tissues. And given a set of arterial blood gases and pH results, determine which acid-base disorder is most appropriate. Okay, now we're just going to cover some basic definitions. So hopefully by now you guys have an idea of what these definitions are anyways through the work that you guys do. I know that you work as MLTs, so we're just going to cover these pretty quickly. An acid is a substance that yields a hydrogen ion or hydronium ion when dissolved in water, whereas a base is a substance that can yield hydroxyl ions, denoted with OH negative. The dissociation constant or ionization constant, or K value, describes relative strengths of acids and bases. And the pKa is a formula that is calculated from that dissociation constant. So it is the negative log of the ionization constant and pH in which protonated and unprotonated forms are present in equal concentrations. And I've got a little note down here that strong acids equal pKa less than 3, and strong bases have a pKa of greater than 9. So you'll see that I have little notes in red here. These are just kind of little carrots of information that you might want to write down. They might be helpful in your understanding. And they also kind of just help you follow along with the lecture so that you're actively writing things down rather than just blindly listening to me if you're listening. A buffer is a combination of a weak acid or weak base and its salt, and this is a system that allows or actually resists changes in pH. And we're going to talk about buffers in a lot of depth today. And why is that? That's because what we're talking about is this homeostasis that our body carries out And part of homeostasis is making sure that we have a good acid-base balance, and buffers are going to help us to achieve that. So, like I said before, acids are chemical substances that can donate protons, or you might also see protons notated as H plus ions in solution, and a strong acid is going to readily give up that proton. A base is what's going to accept the proton from that acid, and strong bases are going to readily accept that proton. So pH is essentially just a measure of those protons, or of those H plus ions. And as you guys already know, a basic pH is any pH that's greater than 7. Neutral pH comes out at exactly 7, and acidic is anything that goes below 7. And pH is a calculation that is the negative log of hydrogen ion activity. And so that's the equation that you see here, the negative log of the hydrogen ion activity. So because we're talking in logs, that means that a decrease of 1 pH unit. So if we go from 7 to 6, that actually represents a 10-fold increase in proton activity. And you can see that there is a table over here that kind of shows you what that really means, what the concentration of the hydrogen or hydroxide ions are, and how that relates back to our pH. So again, like I said, this is important because our body maintains homeostasis. It doesn't want our pH to change much. The body really can't tolerate the pH to really change a whole lot. And so because of that, there's got to be a mechanism in order to keep the pH in the same range, more or less. If the pH gets totally off whack, and by totally off whack, I mean just by maybe a couple tenths of a pH, this can really start to lead to alterations of cells. And in doing so, chemical reactions are not going to perform properly, and that's going to lead to alterations in consciousness. You could get neuromuscular irritability, tetany, and more serious implications including coma and death. So this is something that your body takes very seriously. The range that we're going to be dealing with for the pH, we're mostly going to be looking at the arterial pH. You will also, as a tech, be running venous blood gases, but arterial is kind of the basis that we use for learning, and then venous is off of that. So the arterial range is a pH of 7.35 to 7.45. So that's a very small window for us to get the pH right. And the way that we resist changes in pH within our system is through the buffering capacity of blood. So this is where buffers start to get really important. There are also other mechanisms in which we kind of keep all of this in balance, and we'll talk about those as well. They go hand in hand with the buffering capacity of blood, and they all work together. And that includes some respiratory mechanisms as well as renal regulatory mechanisms. Okay, so this just kind of goes over what acidosis is. Again, if something is acidic, that means that the pH is going to be lower. If something is basic or alkalotic, that means that the pH is going to be higher, and that's what we see here. Okay, so just to touch on some of the buffer systems, the most probably important buffer system that you're going to learn about today is the bicarbonate carbonic acid buffer system. And these are the chemical formulas for each of those, just so that you can start to become familiar with them. So bicarbonate is HCO3 negative, and carbonic acid is H2CO3. We also have what are called the hemoglobin and phosphate buffer systems. There are proteins that are able to act as buffers, the most well-known one being albumin. And then the kidneys also have a way of secreting acid in the form of ammonia in our urine, which is going to help to decrease the amount of protons or the acidity in the blood. Now, like I said just previously, we also have pulmonary and renal systems that work together. So pulmonary, meaning breathing in and out. As you guys know, when you breathe out, you are releasing CO2. And when you breathe in, you are taking in oxygen. So if you wanted to get rid of a lot of CO2 within your system, you would increase your respirations and really try to blow off this CO2. If you wanted to, let's say that you were more basic or alkalotic conditions, then in order to retain CO2 or make things more acidic, then your respirations would decrease. So you would take more slow, deep breaths. The renal system has ways of retaining HCO3, and that's going to increase our pH. Or, like I said before, excrete acids in the form of NH4 in order to decrease the pH. Okay, so again, we're just gonna be focusing on that first acid-base regulatory system that we talked about with bicarbonate and carbonic acid. So, like we said before, all buffers consist of a weak acid and its salt or conjugate base. So here I've just labeled this really important chemical equation. This is super, super important, and you should remember it. I've just kind of labeled who our weak acid is, and that would be the H2CO3, as well as our conjugate base, which would be the HCO3 negative. So just a regular old buffer system. And this buffer system is important for 3 reasons. So you can see that the equation has these like double arrows that are able to go both directions. And that means the body can kind of move this equation in either direction depending on what it needs. So one thing that H2CO3 as the weak acid can do is it can dissociate into carbon dioxide and H2O. And that allows us to breathe off some of that acid, right? So we are able to breathe off CO2 as well as the protons that come along with it as water. And that's just something that we do in our everyday life. We're breathing out CO2 and H2O. And again, you can change that CO2. You can change how much you are respiring. You can breathe faster, or you can take those low, deep breaths, depending on whether you want to retain CO2 or get rid of it. And again, we can alter the HCO3 conjugate base concentration with the kidneys. We can choose to retain that HCO3 if we have especially acidic conditions. Okay, so we had talked about earlier in the objectives the Henderson-Hasselbalch equation and how we can use that in order to assess a particular buffering system. So this is the Henderson-Hasselbalch equation. And basically what we can do is we can eventually just plug in our conjugate base and our weak acid into this formula, what the concentration of those are, and then we can assess our acidity or alkalinity using that equation. So a term that you might see here that may or may not be super familiar is the pKa. And I think there's actually a typo in you guys' notes, so you're going to want to put a little A there. The pKa is basically just a constant that relates back to acid dissociation. And the only thing I really want you to know specifically about the pKa is that the pKa of the bicarbonate buffering system is 6.1. So if we were to plug something into the Henderson-Hasselbalch equation using that really common buffer system that we're going to keep talking about, the bicarbonate carbonic acid buffering system, the pKa that you're going to plug in that case is 6.1. Okay. So these slides, you'll find that they kind of repeat themselves, but hopefully that shows you what is really important for you to know and understand in order to be successful with blood gases. So here we just break down that bicarbonate, or HCO3 negative, is the most important buffer system of plasma, this bicarbonate carbonic acid buffer system. And it is regulated by two separate systems in the body. So not just the respiratory system, but also the renal system. And this ratio here of the concentrations of HCO3 over H2CO3, so the weak acid and its conjugate base, we like to keep that ratio at 20:1. That's really important to note. 20:1 is where we like to keep that ratio, so more base than acid. Now, if you have even more base added to this particular ratio, let's say that you have an excess of HCO3 and you have less H2CO3, what's going to happen to the pH if you have more base? The pH is going to go up, right? And so thus, with the pH going up, we also see that the protons are going to decrease, right? And if we have decreases, this should say alkalosis. If you have decreases in alkalosis, that means that you have less base, right? So if you have less base, that means that you're going to be more acidic. And so your pH is going to be what drops, and your protons are going to increase, because the more acidic something is, the more protons. Okay, and so depending on what causes this increase or decrease, either the lungs or the kidneys will respond in order to restore that 20:1 ratio and to keep the pH within normal limits. And what are the normal limits? 7.35 to 7.45. And you can see that outlined up here too in this nice little diagram. Okay. So we've talked about the bicarbonate carbonic acid buffer system a little bit, and we will talk about it a little bit more, but now that you kind of know the basics and you understand that these can be adjusted in order to maintain that narrow pH window of 7.35 to 7.45, let's talk about a couple of other components that can act as buffers in our system. Hemoglobin is one of them. So hemoglobin is what is the major buffer of red blood cells. So it's going to actually be present in red blood cells, as you know hemoglobin is. And the component of hemoglobin that allows it to act as a buffer is what is called an imidazole group. And that is found in the histidine groups within hemoglobin. So the special thing about these imidazole groups is that they really like to bind protons, which is great because if you have a high amount of acidity, you need something to bind to those protons in order to keep them from disrupting the pH of our system. So if you have any changes that are going to result in a dissociation of hydrogen and you get free protons, we want the hemoglobin to bind those protons. Now, what else does hemoglobin bind? I'm sure you know this. Hemoglobin binds oxygen as well in the form of O2, right? That's its whole goal, really, is to transport oxygen throughout the system to tissues from breathing in the oxygen in our lungs, right? And then the heart is going to pick up that oxygen, distribute it through the blood, and then our tissues are going to benefit from that oxygen distribution. So that's something to kind of take note of here, of how hemoglobin's acting as a buffer. As the O2 binds to hemoglobin, now the protons are going to be released. If the hemoglobin lets go of that O2, now it's going to have room for the protons, and the protons are going to bind the hemoglobin. So that's just something to kind of take note of, is that hemoglobin is not acting as a proton carrier, or it's not holding on to protons as long as oxygen is present on those red blood cells. It's only after the O2 has been released that those protons are going to be picked up by the hemoglobin. Okay. We also have what's called the phosphate buffer system, and this is a very minor buffering system in the blood. You don't need to know much about this other than really the fact that we have protons that are secreted into the tubular lumen by the sodium-hydrogen exchange mechanism that we've previously learned about. And what happens is those protons react with the HPO4. And so as they're doing that, the HPO4 is going to accept those protons or those hydrogens. And when it does that, it's going to convert to H2PO4, which is the acid in this particular equation. So like we had talked about buffers before, we have our conjugate base. That's going to be our HPO4. And then we have our weak acid, and that would be the H2PO4 that's accepting, or that's got the 2 protons. This is then excreted, but it doesn't significantly decrease the urinary pH. That's why we say it's just a minor buffering system of the blood. But it does get some of the acids excreted out of the body through the urine. So that's just one way that we can get rid of excess acid in the body, is to excrete it out with the urine. Like I said before, we also have some proteins that are used as buffers. We'll focus mostly on albumin, and you're going to see another common theme here. So albumin is a protein that can be found in the plasma, correct? And it has 16 imidazole groups from histidine. So it acts actually pretty similarly to hemoglobin in that its imidazole groups are attracted to those protons. So that's what they have in common, and they're going to bind or release protons very similar to the way that hemoglobin does, and that's going to help it to act as a buffer. Okay, so just really quickly, we're going to touch upon this just so that you kind of know why this is so important, and then we'll go back into the acid-base balance. So just really briefly, some of the acid-base disorders that we're going to come across are usually characterized or categorized into acidosis or alkalosis. So if you have acidemia, that means that you're carrying excess acid or proton concentrations in the blood. So your pH is going to be lower than the reference range, right? Because we have excess protons, that means it's an acidic condition, and according to the Henderson-Hasselbalch equation, that means that our pH is going to be lowered, and it's going to be lower than the reference range. You can also have, excuse me, alkalemia or excess base, in which case the pH is going to be greater than the reference range. So greater than that reference range of 7.35 to 7.45. You can have what is called non-respiratory acidosis, and this is where you have a decrease in bicarbonate, and that results in a decreased pH. So it doesn't necessarily mean that you are breathing or that you're not getting rid of enough CO2, but you actually just have a decrease in something basic, and that means that if you have less base, your pH is going to drop. You can have respiratory acidosis, which means that you've got a decrease in the ventilation through your alveoli, and we'll go through how the alveoli ventilate here in just a little bit. But that decrease in ventilation is going to cause you to not be able to blow off that CO2 as adequately, and we like to think of CO2 as an acid, so that's going to result in respiratory acidosis. Your pH is going to be lower than the reference range. Let's write that down. pH is going to be less than reference range. Just as we can have non-respiratory acidosis, we can also have non-respiratory alkalosis. This means that we have an increase for some reason of bicarbonate, which is going to cause an increase in pH as our bicarbonate is basic. And then we can have respiratory alkalosis as well, which means you have an increase in your alveolar ventilation. And this is going to cause you to get rid of too much CO2. So we want to get rid of some CO2, but if we get rid of too much CO2, then that's going to result in creating a basic condition. If we get rid of too much acid, that's going to raise our pH. So let's just write that down. Our pH is going to be greater than the reference range. And as we have briefly described before, the regulation of this acid-base balance can also be acted upon by the lungs and kidneys. So we have our buffering systems, and then we also have our lungs and kidneys. So if you haven't figured it out by now, ventilation, breathing in oxygen, breathing out CO2, is going to affect the pH of the blood, and that's because we think of CO2 as an acidic component. So CO2, or O2, as we breathe it in, it diffuses from the alveoli into the blood, and then the CO2 is going to diffuse into alveoli from the blood and be eliminated via ventilation. And we'll look at that more closely through a figure here in just a minute. The result of that is a minimal change in the proton concentration between venous and arterial circulations. So that's what's really important, right? Is we want to be able to exchange those protons from one carrier, if you will, to another without causing a huge change in those proton concentrations that our body is going to notice that's going to result in a pH change. The kidneys, on the other hand, are going to do this a little bit differently. The way that they're going to regulate the acid-base balance is by excreting acids or bases, and I should say, or retaining bases, such as HCO3. That's something that our kidneys are known to do as well. So the HCO3 can be reclaimed from the glomerular filtrate, and that's going to prevent excessive acid gain in the blood from loss of HCO3 in the urine. So if we're losing too much base or HCO3 out of our urine, that's going to cause an acidic condition within the blood. So what we can do is we can, well, I guess we don't tell them, but our kidneys automatically decide that it's a good idea to just start retaining some of that HCO3, retaining some base to resist acidic conditions. Okay, so I've said this before, but something that you should be kind of putting together by now is that you can think of CO2 as an acid, and you can associate it with respiratory imbalances because you're breathing out CO2. Similarly, you should be thinking of HCO3 or bicarbonate as a base, and this is something that you're going to want to associate with metabolic imbalances. Why? Because our kidneys have the ability to excrete or retain HCO3 in order to minimize major pH changes. So that one, anything related to the kidneys, we're going to refer to as a metabolic imbalance. Anything related to breathing in and out, naturally, is going to be a respiratory imbalance. Okay. So let's take a look at this pulmonary system regulation and how that kind of works. So again, here comes this really important equation, right? This is the bicarbonate carbonic acid buffer system that we just keep coming back to. So if you have in the middle, you have your weak acid, the carbonic acid, and then you've got your double-sided arrows, meaning that the equation can go back and forth depending on what the body deems necessary. Your weak acid can either dissociate into a proton and bicarbonate, or it can dissociate into CO2 and H2O, CO2 being an acid that we're going to breathe out. Okay. So if you have an acid increase, if the system starts to contain too many protons and our pH goes down, then what's going to happen is we're going to go here and our protons are going to combine with bicarbonate. Those are going to create this weak acid. That weak acid is then going to dissociate and become CO2 and H2O. And then we can release that CO2 through our respirations, and that should help us to get rid of some of the acid that has been increasing within the system. I hope that makes sense. If you have an alkali increase or your pH is going up, one way that the body can compensate for that is to retain your CO2. So CO2 is acidic, right? And things are getting too basic. So what can we do to alter that pH? We can start to retain some acid. So to do that, your body's going to know that it needs to respire less. So you're going to decrease your breathing activity. Okay. How does the renal system regulate? Again, we've briefly talked about this. The kidneys can choose to retain bases or they can choose to excrete more acid. So if they're going to retain the bicarbonate, if you happen to need more basic conditions, if they're going to retain bicarbonate, that's going to happen from the proximal tubule of the kidneys. If you need to excrete more acids, that's going to happen via the distal tubule. And so that can happen in a couple of different ways. We can secrete protons specifically by using the bicarbonate carbonic acid buffer that we've been talking about so much. We can combine protons, and we're going to look at this actually on the next slide, but it never hurts to go over twice. You can combine protons with NH3 to make NH4, which is secreted or excreted out of the urine. And we can have an H2PO4 phosphate buffer as well. So that was what we talked about previously, where we had HPO4 and we were having it pick up a proton to make H2PO4, which is then excreted out of the urine. So both of these can be excreted. Okay. So, like we'd previously discussed just now, you can use ammonia as a way to release acid out of the body by excreting it from the urine. Okay, so now we're going to talk about the distribution of gases in the body. So we're kind of moving now from exactly how this acid-base homeostasis works into how do we really distribute oxygen and CO2 throughout the body, and how do they kind of keep each other in check. So we're going to look at the transport of oxygen, we're going to look at the transport of carbon dioxide in the blood, and then we're going to look at the hemoglobin oxygen dissociation curve. So to do this, to understand this, the first thing you need to understand is about partial pressure. So partial pressure, anytime that you see like a PO2 or a PCO2, all that's telling you is it's the partial pressure. And this just comes into play when you're dealing with gases. So when more than one gas is present in a container or within the body, which of course is always going to happen. You're never going to have 100% of one gas and 0% of the other. The pressure exerted by one gas is called the partial pressure. So this just kind of shows us the composition and partial pressures of gases in a dry air atmosphere. So these are just any number of gases. Okay, so now that we kind of know what the partial pressure concept is, and we can understand that partial pressure is really just a way of expressing how much of a particular gas is, how much pressure a particular gas is exerting in a container or a body, we can start to apply that to how we transport CO2 and oxygen throughout the body, and how that transport really depends on partial pressure. A lot of this is going to rely on the fact that gases travel from high concentrations to low concentrations. Pretty basic concept there, and you'll see how that applies here. So I left a little box here saying that we were going to draw something out in the video lecture, and as promised, we are going to draw something out, but after thinking about it, probably is not going to fit into this little box. If you can fit it into this little box, that's awesome. I will not be able to. So I'm just going to add a page to the notes, and I'm going to draw it out on a full page or maybe even a couple of pages. And I would really highly suggest that you draw this along with me or follow along with me, but probably draw it along with me, because this is really going to help you to hammer in these concepts. It really helped me to understand them better, to be able to use them in the lab. So I would really recommend that you draw this with me. So if you don't have a blank piece of paper, you're going to want to get one now. And you can pause the video and grab a blank piece of paper or a couple pieces of paper, some colored pencils if you have it, and let's get to drawing this thing out. Okay, so we're going to start with a blank slate here, and we're just going to write the definition of partial pressure up at the top. So partial pressure, as discussed previously, it's very similar to the concentration of a gas, the amount of pressure that a gas is putting on a system. The higher the partial pressure, the larger amount of gas present. So it's proportional to the concentration. Okay, so if we were to draw a guy, and forgive my cartoon drawing skills, if we were to draw a very serious looking guy, I don't know, with apparently an egg-shaped head. Oh, that wasn't so bad. If we were to draw a guy and we were to draw his respiratory airway down into his lungs, and his diaphragm. Okay, now we can start to talk about how air moves around. So let's talk about what is present in our atmosphere, what we're breathing in terms of the partial pressure of oxygen and CO2. So in the atmosphere alone, our PO2 is going to be around 160 millimeters of mercury, or mmHg. And this is the only time I'm going to write millimeters of mercury, just for the concept of saving space. But rest assured, any of the partial pressures that we're going to show on this diagram are going to be denoted in millimeters of Hg, or millimeters of mercury. So the partial pressure of O2 in the atmosphere around 160 millimeters of mercury, whereas CO2 is actually only around about 0.3. So that's what we're breathing in. And as you know, we're going to eventually transport the oxygen to our tissues to be used. Okay, I'm going to use some colors here to hopefully try to make this pretty clear. So as you can see, the PO2 is a lot higher in the atmosphere than the PCO2. Okay, so we're going to breathe that in. And it's going to go down into the lungs. Now, in turn, what are we breathing out? We're breathing out CO2, right? Mostly. We're also breathing out some oxygen. But CO2 is what we think of when we think of breathing out. So the partial pressure of what we are breathing out, we're actually going to have a PO2 of around 120. This is surprisingly high, right? And then we're going to have a partial pressure of CO2 of about 27. This is what we breathe out. So just kind of a quiz question, I guess, or a fun trivia question. If we're breathing out that much CO2, what's happening to it in the atmosphere? Because it's going from 27 to 0.3. What do you think is happening? I'm sure you've guessed it. The plants, right? The plants in our world are taking that CO2 and using it for their cellular respiration and in turn giving us oxygen. So they're taking the CO2, they're doing some stuff with it, which we're not going to go into today. And then from that CO2, they're making oxygen for us to breathe in. Okay, so let's keep drawing. So now that oxygen and CO2 that we are breathing in is going to go down into the lungs, right? And it's going to go into the alveoli. So I'm just going to draw kind of what a zoomed in picture of the alveoli would look like. And it's probably going to be pretty bad. Okay, so we've got some alveoli there. Each of these little individual circles being the alveoli. And then we've got the pulmonary artery, which is going to take deoxygenated blood and it's going to deliver it to the alveoli. So this is our pulmonary artery, and it's going to deliver deoxygenated blood to alveoli. Now, when this same blood leaves the alveoli, it's going to get oxygenated, right? Because the oxygen is in the lungs. And so it's going to get oxygenated, and the blood's going to leave the alveoli via the pulmonary vein. So that's what we're showing in red here. This is the pulmonary vein in which oxygenated blood leaves the alveoli. Okay, so there's our zoomed in picture of the alveoli. Now let's zoom in even further into one singular alveolar sac. And let's look at what's happening there. So oxygen is coming in to the alveoli. And we know that CO2, when we breathe out, is going to be leaving the alveoli. Now, the partial pressure of oxygen and CO2 in the alveoli is going to come to be very important. So the partial pressure of O2 in the alveoli is about 100 millimeters of mercury, whereas CO2 is going to be about 40. So we're going to see a lower concentration of CO2 in the alveoli than what we see of O2. So let's draw that out just in a little color-coded cartoon. Got PCO2, and then we've got more oxygen than we have of CO2. And this is the alveoli. Hopefully I'm not going too slow. Okay, so how does the oxygen make its way out of the alveoli? We talked about this just a second ago. It's going to leave via the pulmonary vein, right? So let's just draw that pulmonary vein. And this is how the oxygen is going to travel, is through that pulmonary vein onto tissues. Now, this is where the partial pressure becomes important, because the partial pressure in here is going to be 100 for oxygen, 40 for CO2. Okay, so same as before, we've got a higher concentration of oxygen and a lower concentration of CO2, because our whole point here is to transport oxygen to the tissues this way. Now, what about the CO2? Now, let's look at the pulmonary artery, which takes deoxygenated blood to the alveoli. So that it can be oxygenated. So we have here our pulmonary artery. Now, in the pulmonary artery, the partial pressure of oxygen is only 40, whereas the partial pressure of CO2 is actually higher. It's going to be 46. So, as we talked about before, gases are going to move from high concentration to low concentration. So the CO2 in this case here in the pulmonary artery is 46, where it's only 40 in the alveoli. So that's why the CO2 is going to travel into the alveoli, because it's moving from high concentration to low concentration. So the CO2 is going to go from the pulmonary artery into the alveoli. Hopefully that makes sense. Whereas the oxygen is going to travel out to tissues via the pulmonary vein. So let's just draw that out in a cartoon over here in the pulmonary artery. For once, we've got more CO2 than we've got of oxygen. Okay, now this is where things start to change a little bit. I'm going to see if I can add another page to this, because I'm drawing even bigger than I had originally thought. Did that work? Nope, that added it below, or above. Let's see if we can, nope. Hang on. Okay, I'm going to see if I can't move this. Sorry. Just minor delay. At the very least, we can shrink it. That's helpful. Oh, nope. Okay, so sorry for the technical difficulties here. I'm still learning this thing. Okay, so now what we're going to look at is how this connects to the heart. So I'm just going to kind of make a connection here. So we've got the alveoli above, and then here we've got our heart. So this oxygenated blood is going to travel from the pulmonary vein into the heart, and then the heart is going to distribute that blood into tissues. And that's going to happen via the systemic artery, which is carrying oxygenated blood. So again, our partial pressures are important here. In the systemic artery, our partial pressure of O2 is 100, and our partial pressure of CO2 is 40. So again, we're carrying oxygen, so we've got a higher concentration of oxygen than we have of CO2. And those are going to be delivered to the tissues. So let's just draw some tissue. Hopefully this remotely looks like tissue. Now the partial pressure in tissue of oxygen is 40 millimeters of mercury, and the partial pressure of CO2 is going to be at least 45 millimeters of mercury. Okay, so now we're starting to see where we've got a difference in our partial pressures. So again, gases are going to travel from high concentration to low concentration. And so because of that, we're going to see oxygen traveling from high to low. So it's going to be traveling from that systemic artery where it's partial pressure of 100 to a lower concentration in the tissue that has a partial pressure of 40. So let's just draw those partial pressures again in the tissue, what those look like. So that's why oxygen is going to be released into the tissues. Now, as a byproduct of metabolism of the cells found within tissue, what do you think we're going to produce? We're going to produce some CO2. Okay, so let's hop over to the other side of this, and we're just going to draw out our systemic vein. Systemic vein. Let's label that. And the systemic vein is what is going to carry deoxygenated blood. Carries deoxygenated blood. Back to, you guessed it, the heart. So it's going to take deoxygenated blood and it's going to take it back to the heart. Okay. So like we had said before, the byproduct of tissue metabolism is going to be CO2. That's going to create an acid buildup, correct? If we have a buildup of CO2 within the tissue. So we want to get rid of that acid in order to maintain that pH homeostasis within the body. So we need to carry that acid back up to the heart and then up into the lungs so that we can breathe that CO2 out, right? So CO2 is going to travel out of the tissues and into the systemic vein through capillaries and then back to the heart. Now, why does this happen? Again, the CO2 is going to travel from high concentrations to low concentrations. So if we're getting a CO2 buildup, as a result of tissue metabolism, or metabolism of cells within the tissue, then we're going to get a higher concentration of this PCO2. If we know that the partial pressure of oxygen is 40 in the systemic vein and the partial pressure of CO2 in the systemic vein is around 46, then that high concentration of CO2 is going to travel to lower concentration, hitch a ride through the systemic vein to the heart, and then once it makes its way to the heart will then pump that deoxygenated blood back into the lungs via the pulmonary artery. And then the cycle begins again, and we breathe out that CO2. Okay, I hope that that made sense. I hope that that shows you that the partial pressure is really what's going to dictate where gases move. So any gas is going to move from high concentration to low concentration, and that's what's going to help to push oxygen around the bloodstream and push CO2 out of the bloodstream, out of the lungs, so that we can breathe it out. Okay, so now let's focus on a little bit more specifically the oxygen transport. So as you guys already know, most oxygen in arterial blood is transported to tissue by hemoglobin. And just really quickly, blood hemoglobin can exist in one of four conditions. One being oxyhemoglobin, which is just the form of hemoglobin that is reversibly carrying oxygen around. So the oxygen can be dropped off to tissues or it can be picked up and taken around. The deoxyhemoglobin is hemoglobin not bound to O2, but it's open to binding to O2 anytime the O2 is available. So it's just not currently bound to the O2. Carboxyhemoglobin is hemoglobin that is bound to carbon monoxide. So I'm sure you've heard about that, how carbon monoxide just has a really high affinity for hemoglobin, or hemoglobin has a high affinity for carbon monoxide. And we'll talk about that more in the next lecture. Methemoglobin is hemoglobin that's unable to bind oxygen, and that's because the iron that is within the heme molecule is in its oxidized form rather than its reduced form. So the iron itself is in the Fe3 plus form rather than the Fe2 plus form, and because of that, it's not able to bind oxygen. And that is problematic, is it not? If your hemoglobin's not able to work properly, it's not able to bind to oxygen to transport oxygen throughout the body, that's an issue. So both of these kind of problematic forms of hemoglobin, we're going to call those dyshemoglobins. Okay, and here is the hemoglobin oxygen dissociation curve that is so important. So I've written some notes on the dissociation curve. Some things to notice about the curve is that it is sigmoidal or kind of S-shaped, and that's because eventually at the top here it's going to plateau. And this plateau just signifies that no matter how much more oxygen is present, hemoglobin is essentially fully saturated. So it's not able to pick up any more oxygen. So adding more oxygen is not going to increase the amount of hemoglobin that is bound to oxygen once you get to this plateaued form. So that's what kind of makes it the sigmoidal shape. That plateau is important. Some factors that affect the hemoglobin oxygen dissociation curve. So let's just think about what that even means. What we're talking about here is the affinity that hemoglobin has for O2 molecules. Or how likely is the hemoglobin to grab onto that oxygen, and how likely is the hemoglobin to let go of that oxygen. So factors that can affect that are temperature. Of course, temperature affects any kind of reaction. pH, of course, this is why it's so important, or one of the many reasons why it's so important for us to regulate pH. The partial pressure of CO2, and 2,3-diphosphoglycerate concentrations, or 2,3-DPG. Okay, so how can we look at this? Let's look at this bottom curve here. So in this bottom curve, what we're showing is that the affinity for oxygen by hemoglobin is decreased. So hemoglobin is more likely to let go of oxygen in this particular curve. So what kind of conditions are these? These are conditions that are lower in pH, higher in CO2, higher in 2,3-DPG, and higher in temperature. The perfect area that is described by these conditions are tissues. And that makes sense, right? Because we want hemoglobin to let go of oxygen in order to give oxygen to those tissues. That's its job, is to transport oxygen to tissues. So this is where we want hemoglobin to actually dissociate from the oxygen. What we see in the middle here, this is kind of our standard. This is middle ground. Now, if we describe this particular curve in comparison to this curve, what we would call this is a right shift. Similarly, in talking about this particular curve compared to this middle curve, this would be called a left shift. So the left shift, what we're saying here is that the affinity for oxygen by hemoglobin is higher. Hemoglobin is more likely to hold on to that oxygen. What kinds of conditions might this be in? We've got an increased pH, we've got a lower amount of CO2 pressure, we've got a decreased amount of 2,3-DPG, and we've got a lower temperature. So that's going to result in hemoglobin holding on to oxygen more tightly than it normally would. And the perfect place for this to occur is in the lungs. Now that makes sense too, because we want hemoglobin to pick up oxygen in the lungs so that the oxygen can then be transported to tissues, correct? So we want it to hold on to oxygen in the lungs. Okay, hopefully that makes sense. So you should be able to look at a hemoglobin oxygen dissociation curve and describe what the different conditions are if you have a right shift or a left shift, and what is an example of an area where those conditions might occur, and what does that shift mean in regards to hemoglobin affinity for oxygen. Okay, let's talk about oxygen and gas exchange briefly, and then we'll be finished with this beast. So, When I talk about oxygen and gas exchange, what I'm discussing is oxygen and carbon dioxide exchange. So kind of what we talked about earlier with that giant diagram that we drew, but just going into it at kind of a different angle. So there are 7 different conditions that are necessary for adequate tissue oxygenation. So for oxygen to be delivered to tissues in such a way that the tissues have plenty of usable oxygen to aid their cellular respiration. So first of all, you've got to have available atmospheric oxygen, right? You've got to have oxygen in the air to breathe in. You've got to have adequate ventilation. You need to have an adequate gas exchange between the lungs and arterial blood. That's what we went through in that diagram. You've got to have a good gas exchange between the lungs and arterial blood. You've got to have a hemoglobin that's able to pick up oxygen. So that's relating back to the hemoglobin oxygen dissociation curve. You've got to have an adequate amount of hemoglobin. So individuals who are anemic, right, and have a lower amount of hemoglobin are going to have more issues with oxygen exchange. You've got to have adequate transport of those tissues. And then you've got to be able to release that oxygen into the tissue. So you want the hemoglobin to be able to decrease its affinity once it gets to tissues and be able to let go of that oxygen upon delivery. Some common factors that you will encounter, some physiological factors that really influence the amount of oxygen that's able to move through the alveoli and into the blood and onto tissue. Of course, destruction of alveoli is going to influence the amount of oxygen that's able to move through the alveoli, right? They've got function or form equals function. So if the form of the alveoli is destroyed, then the function of the alveoli is not going to be very good. Pulmonary edema, again, if you have some sort of blockage in that area, then you're not going to be able to transport oxygen properly. And pulmonary edema is the idea of having liquid in that area, so that's going to cause a blockage. Any kind of airway blockage. If you have inadequate blood supply to tissues, then you're not going to be able to transport oxygen to those tissues if blood's not able to travel there. And of course, the diffusion of CO2 and O2. That's really the big one. Okay, here are some terms that you will come across working in the lab that will help you to assess a patient's oxygen status. These are something that we look at when we run, say, a venous or an arterial blood gas. If in your particular hospital the respiratory therapists are running these blood gases, then this might not be something that you encounter very often. Oxygen saturation, or SO2, that is how much oxygen is bound to hemoglobin compared to the total amount of hemoglobin capable of binding to O2. And we like to see that number around 95%. That's definitely good. If we start to dip below 90%, that's not good. So we want to see that number above 95%. Fractional oxyhemoglobin is the ratio of the concentration of oxyhemoglobin to the concentration of total hemoglobin. We can look at trends in oxygen saturation. So trends in oxygen saturation by pulse oximetry. So you guys have seen those little machines that get slipped on the tip of someone's finger. Sometimes on babies, they're wrapped around their foot. That is just a transcutaneous or through the skin way of looking at oxygen saturation. And then finally, oxygen content is the total amount of oxygen in the blood. So we're talking the sum of oxygen bound to hemoglobin in addition to the amount of oxygen that is dissolved in the plasma. Because there's always going to be a certain amount dissolved in the plasma. So in this case, we are looking at the total amount of oxygen in the blood, not just that blood that is bound to hemoglobin. Now, carbon dioxide is transported in the blood in 5 different forms. So it can take on a lot of different forms. There's a lot of different ways to assess carbon dioxide. So carbon dioxide can take the form of bicarbonate, right? So the HCO3, through carbonate, carbonic acid. We can have dissolved carbon dioxide in the plasma, and we can also have carbon dioxide binding to proteins within the plasma, forming carbamino compounds. So 5 different ways. Now I want to just briefly talk about something called the chloride shift. And this is how CO2 is going to move from the tissue, right? So remember, it's expelled out of the tissue as a byproduct of cellular metabolism into the plasma and also into the red blood cells. So carbon dioxide diffuses out of the tissue where it's produced. So right here, we're looking at the tissue. And it's going to go a couple of different ways. It can go into the plasma, and it can go into red blood cells via surrounding capillaries. In the plasma, this is where we're going to see dissolved CO2, or we're going to see CO2 combining with proteins to make those carbamino compounds that we talked about before. Now, when the CO2 goes to red blood cells, what we're going to see happening is the CO2 is actually going to bind with water, and that's going to form carbonic acid, and then that carbonic acid is going to dissociate into the bicarbonate and H+. Okay, so that equation should look familiar. That's really similar. In fact, it's exactly the same to the bicarbonate carbonic acid buffering system that we talked about at the beginning of the lecture. So you should recognize that. So once we get this whole chemical pathway occurring, what you're going to see is we're going to have a buildup of bicarbonate now in the red blood cell. Again, high concentration to low concentration, the bicarbonate is going to travel out of the red blood cell into the plasma because it's in high concentrations in the red blood cell and low concentrations in the plasma. So it's going to diffuse out of the cell and into the plasma. And when that happens, it's actually going to trade places with chloride. So chloride and bicarbonate have kind of an exchange system where chloride comes in and bicarbonate comes out. And this is going to be important in our next lecture when we're talking about different causes of alkalosis and acidosis. Chloride can be involved in this. Okay, so what this trade of chloride and bicarbonate is termed is a chloride shift. Okay, so just put that one in your pocket for our next lecture. Okay, we have made it to the last slide. Pat yourself on the back. This has been a long one. I know it's difficult and I know that it's kind of tedious, but you've made it to the last slide, so stick with me for the last 45 seconds. Okay, transport of carbon dioxide now in the blood to the lungs so that we can respire that CO2 out. So we've discussed CO2 going from as a waste product in tissues into red blood cells and plasma. Now we're talking about let's take that CO2 from the blood and transport it to the lungs so that we can breathe it out. So in the lungs, the process is actually reversed. We breathe in oxygen. That oxygen has an exchange system with protons that are carried by the hemoglobin. So this is that hemoglobin buffer system. Hemoglobin is going to let go of the proton to grab onto the oxygen. And those protons are going to combine with the bicarbonate in the blood to make H2CO3 or carbonic acid, which will then dissociate into H2O and CO2. That CO2 can then diffuse from a high concentration to a low concentration in the alveoli, and then we breathe it out. We breathe out CO2 and H2O. And that, you guys, is the end of this lecture. If you have any questions at all, please feel free to email me. My email is kwilling@uyo.edu.